Assalamualaikum warahmatullahi wabarakatuh, and greeting to all viewers. Today I'm Muhammad Johari Ibrahim from Faculty of Medicine, University Technology Mara. On behalf of our group, would like to share our current innovation entitled The Development of Radio-Resistant Cell (EMT RRMGI) to Gamma Ray Irradiation from EMT6 Mouse Mammary Carcinoma Parental Cell for Understanding Cancer Radio-Resistant Mechanism. There are no definite explanation how radiation incident is occurred, but it is believed that certain cells are able to escape the radiotherapy treatment and form the new cell population. So it becomes a major question how certain cells can escape the ionization treatment, and many hypotheses has been postulated. For example, increased cell cycle checkpoint that enable the cell survival, increased cell proliferation post radiation treatment, increased formation of cell stemness, and that have the cell stemness characteristic, and formation of angiogenesis, which is the new blood vessel in the tumor microenvironment. So the objective of our study was to develop a radio-resistant EMT mouse mammary carcinoma to radiotherapy, which is the gamma ray irradiation, for further study to investigate the mechanism of radio-resistant cancer cell, focusing on breast cancer cell. In this study, we use in vitro model using EMT6 mouse mammary carcinoma cell line using two approaches, where the direct irradiation from low to high dose of fractionated irradiation. For direct irradiation, we use 2 gray for until 8 gray, while 0 gray serve as control. For fractionated irradiation, we give 2 gray per cycle to mimic the clinical setting, where there are sham control. For each cycle, we do the clonogenic assay to measure the survival fraction of the cell after radiotherapy, while the remaining of the cell is grown for the next cycle irradiation with 2 gray, and then the same step occur. Okay, this step is repeated until cell resistance is developed. How we measure cell resistance is developed? Where the survival fraction is plateauing. This slide shows the pre-summary of clonogenic assay. Clonogenic assay is a gold standard to measure the effect of radiation, where after irradiation, we will seed the cell into 10-liter petri dish, and then we will let them to grow to form the colony between 8 to 10 days. And then the survival fraction were calculated, where the number of colony were divided into the number of cells seeded, and cell survival curves were plotted. Okay, in figure 1a, you can see there are two lines. One is black line, and then the second one is the dotted line. The black line refers to the direct irradiation from low to dose. You can see that here, and when at the dose 8 gray, majority of the cell is dead. Okay, while using the fractionated up to 4 gray, okay, the survival fraction is reduced, and then after third and fourth cycle, the survival fraction increase back, and then after 5 up to 8 cycle, the survival fraction is plateauing. So here we believe that radio resistance already developed, and then to confirm this, okay, we irradiate cell from using low to high dose in single fraction, and then here we can see that the blue line is the parental cell, the black line is the radio-resistant cell. So radio-resistant possesses a high survival fraction that prove radio-resistant already developed after 8 cycles, which is 16 gray of radiation. As a conclusion, we have successfully produced radio-resistant cell called EMT-RRMGY cells using fractionated radiation at 2 gray per cycle after 8 cycle, which is the total dose 16 gray. Okay, there are three main outcomes from the study. First, we have successfully granted copyright from MyIPO regarding the production of the radioresistance cell, and then we already published a conference journal this year. And then third, we have research collaboration with University Putra Malaysia and then University Kebangsaan Malaysia. Okay, for the novelty of our project, we have produced a radio-resistant breast cancer cell, EMT6 RRMTI, that can be used to investigate the mechanism of cancer cell resistance. And then the usefulness of our product is this radio-resistant cell can be used to investigate the radio-resistant mechanism to radiotherapy to produce radio-resistant cancer cell. And then this can improve the effectiveness of treatment for breast cancer patients, thus improve the quality of life as well as the budget spent by government for cancer treatment. Okay, last but not least, I would like to thank our group. Fatiha is my PhD student, Dr. Muhammad Khalis, a medical physicist from UPM, and then Dr. Narim Abdul Dasani is a molecular biologist from our faculty. Thank you.